Hello everyone, students of AP Biology. This is the first session for revision for the AP final exam. I'll be dividing the revision into different sessions, and I'll be starting with the cell organelles and compartmentalization. So in this lesson, we are going to have a preview on the cell organelles, their function, and a brief look at their structure. So in general, cells are structural and functional units. It means they have a certain structure and they perform different functions. Now, you know that there are two main types of cells, prokaryotic and eukaryotic cells. And the main difference between these two cells is two. The first one is the presence or absence of a nucleus, but not only nucleus, membrane-bound organelles that have membranes. All cells, despite all of their types, they have the following. They have cell membrane, cytosol, chromosome, and ribosomes. So ribosomes are not membrane-bound organelles. Why? Because they exist in all cells. They are only proteins and ribosomal RNA. So the main difference between prokaryotes and eukaryotes, as we mentioned, is the presence of a nucleus and the presence of membrane-bound organelles, where they are absent here, but DNA is there, ribosomes are there. Examples on prokaryotes are bacteria and archaea. Examples on eukaryotes are protists, which are single cell, fungi, which can be single or multicellular, and animals and plants. To start with the organelles, the membrane-bound organelles, which are found in eukaryotic cells, are classified based on their shape and based on their function as well. Starting with the nucleus or the nuclear envelope, endoplasmic reticulum, which we have two types, rough and smooth, Golgi complex or Golgi apparatus, lysosomes, vesicles and vacuoles, and the plasma membrane, and the energy organelles, mitochondria and chloroplast. Why we call them energy organelles? Because simply those are the organelles responsible for generating the cell's energy in both animals and plants, knowing that chloroplasts are only present in plant cells. Compartmentalization. The concept of compartmentalization is an important one to allow the cell to perform different functions at the same time. So imagine you are a company or a firm, and this company performs a lot of tasks. You cannot do all the tasks by the same people in one room. You need to have different departments. The same organization of a school or an organization or a firm can be described in the cell by compartmentalization. There are different compartments in the cell. Those compartments are surrounded by membranes. We are speaking about membrane-bound organelles that perform different functions. Now, the importance as well of compartmentalization is giving a space, a specific area for every organelle to perform by its own, and this will increase the surface area for reactions. For example, the reactions for cellular respiration takes place, the reaction of cell respiration, which is breaking down glucose to make energy, it takes place in mitochondria, only in mitochondria. And when we look at the structure of the mitochondria, you are going to see that mitochondria has folded inner membrane. Let's go to the mitochondria quickly. So the inner membrane of the mitochondria is folded, this one here. So this will increase the surface area. As a result, there will be more reactions. Because later on in next chapter, we are going to study about the cellular respiration chemical reaction, and you will see that this reaction takes place in the membrane. So since I have folds here, it means we have larger space for the reactions to take place. So compartmentalization, it helps organizes the reactions that take place and increase the surface area, as well as preventing interfering reactions from occurring at the same location, so that reaction doesn't happen at the same time and they don't interfere. Speaking about eukaryotic cells, it's good to recall that plant cells have chloroplast, central vacuole for storage, cell wall for protection, and plasmodesmata. Plasmodesmata, those are modified structures that are connected to the plasma membrane, and they allow the connection or the adhering of cells beside each other. So that's why if you look at plant cells under microscope, you won't see spaces. All cells are connected together. Those organelles or parts are not in animal cells. At the other hand, lysosomes, centrosomes, and flagella are in animal cells but not in plant cells. It's good to distinguish between these different parts. So let's speak a little bit about each organelle, and here I'm going to explain very briefly so that I can cover all the material in a shorter period of time. Let's start with the nucleus. It's the place where chromosomes, DNA, genes, genome, whatever you can call it, all the hereditary information is there. Okay? The nucleus is made of different parts. We have the nucleolus. This is the dark region here. Its main function is to produce ribosomal RNA. Then you have the space nucleo, like the cytoplasm, we call it the nucleoplasm, where we have DNA and the chromosomes. Then we have the membrane, which is double membrane, not one, double membrane. So it's double phospholipid bilayer. And within this membrane, there are the nuclear pores that allow exchange between the nucleus and the cytoplasm outside. And those pores is exactly where messenger RNA or the code is going to leave into the ER in order to start the process of protein synthesis. As we said, nucleolus is to make ribosomal RNA, which are part of the ribosomes that make proteins that can leave through nuclear pores. Now, since we are speaking about ribosomes, let's recall protein synthesis. Protein synthesis is one of the most important functions that the cells undergo, which is making protein. It all starts in the nucleus. It all starts with the DNA being transcribed into messenger RNA. Messenger RNA is a copy of DNA, but it is single strand with some changes. Instead of T, we have U, and instead of double strand, it's one strand. And one more thing about messenger RNA, which is so important, messenger RNA is created in the nucleus, but it has the ability to move outside the nucleus to go to the cytoplasm. Why? In order to transcribe or take this information, which is stored inside, and take it outside. Okay, in order to start the process of making protein. So the first step of making protein or protein synthesis is DNA is transcribed to messenger RNA, then messenger RNA exiting or leaving the nucleus through the nuclear pores to where? To the endoplasmic reticulum. And when we speak about endoplasmic reticulum, we speak about two types of ER. We have the smooth ER and the rough ER. Now, the rough ER, it contains ribosomes and it is located directly around the nucleus. So rough ER is connected to the nucleus, and it is rough because it has ribosomes. And ribosomes are the organelles that are going to host the messenger RNA. So this is the third step after messenger RNA left the nucleus. They are going to go or become installed in the ribosomes in the rough ER. And this is where protein synthesis starts by a process called translation. What is translation? It's translating the messenger RNA into protein by binding amino acid together. You know this information from chapter 1. So, going back here, the messenger RNA transcript must be then modified before leaving the nucleus. Then in the cytoplasm, it will go through the ribosome. This is the central dogma of biology, where DNA is converted to RNA. RNA is used to make protein. So ribosomes, as we said, they are not membrane-bound organelles. They don't have membranes. That's why they are present in all cells, and all cells need ribosomes because cells cannot survive without making proteins. Bacteria cannot survive without certain enzymes, and those enzymes are proteins. For this reason, all cells have ribosomes. The main function is to make proteins. And there are two types of ribosomes, either attached to the ER or they can be floating freely in the cytoplasm. So they can be in the cytosol or the cytoplasm, or they can be attached to the ER. Now, one difference when it comes to the function, proteins produced here generally function only within the cytosol. So ribosomes that are free in the cytoplasm, they produce proteins that function within the cell, for example, enzymes. Whereas proteins that are produced in the ER, those proteins will be sent outside. So if proteins are, for example, food that is being served in or prepared by a restaurant, the food that is prepared by the free floating ribosomes is going to be served inside, dining in. And the proteins that are going to be produced by the rough ER are the delivery proteins that are going to be sent outside the cell. And here comes the different steps of sorting, modifying, and packaging proteins that happens in the Golgi apparatus. Okay, so after proteins are produced here in the rough ER, they are transported through vesicles to the Golgi apparatus, which is another membrane-bound organelle at the edge of the cell. Before we continue about the protein protein synthesis process, let's speak a little bit about the two types of endoplasmic reticulum. As we said, we have the smooth and the rough ER. So the rough ER, it mainly makes protein, full stop. The smooth ER, it has different functions. It doesn't make proteins because there are no ribosomes, and that's why it's smooth, but it produces or synthesizes lipids. Moreover, metabolizes carbohydrates, and one of the most important functions of smooth ER is detoxifying the cell, removing alcohols and drugs and all toxic materials. That's why people who take alcohol, drink a lot of alcohol, they tend to have large number of smooth ER in their liver cells, because liver, it's where the cleaning or detoxifying of blood takes place. So that's why they have high number of smooth ER in their cells, because they need to clean a lot of toxins. So again, this is smooth and rough ER. Now, Golgi complex. Going back to protein synthesis. So I'm going to repeat this from the beginning, from the nucleus, DNA turning into messenger RNA leaving the nucleus through the nuclear pores, then going to the rough ER, to the ribosomes, where proteins are made. After proteins are made, they are transported to the Golgi apparatus through vesicles. Now, the Golgi apparatus is made or is divided into two sides based on its direction. The one which is facing the nucleus, I'm speaking about this one here facing the nucleus, we call it the cis Golgi apparatus, or the cis side. And the trans one is facing the membrane. So this is trans, this is here cis. Now, what will happen? The cis side, which is closer to the nucleus, is going to receive the vesicles coming from the ER that contains proteins. So imagine a vesicle like a submarine transporting people underwater. So the vesicles that are produced in the ER are going to transport the protein into the Golgi apparatus, mainly into the cis phase of the Golgi apparatus. Now, what happens there? In the Golgi apparatus, proteins are modified. What does modification of proteins mean? It means that proteins are labeled with carbohydrates or glycoproteins. Those carbohydrates can act as indicators for proteins in order to tell where those proteins are going. So they are like labels. So imagine speaking about a restaurant, you want to send a delivery, you add on the box of the food, where after you wrap the food or you package it, you add the destination. This is done in the Golgi complex, where proteins will take a label there. After that, proteins are going to move through vesicles from the trans phase to outside, because they are going to be transported outside by exocytosis. So the main function of Golgi complex is to receive transport vesicles from the ER, modify the material, not only proteins but even lipids, sort the material, package them, add tags, and release them by exocytosis. Because the trans phase is close to the membrane, they will release them. One specialized vacuole or vesicle in the cell, lysosome. Lysosome, it's the factory of enzymes. And enzymes are needed for a lot of functions. One of the most important enzymes in the body are hydrolytic enzymes that break down material. For example, breaking down proteins into amino acid, breaking down carbohydrates into glucose, breaking down lipids into fatty acid and cholesterol. So those enzymes are produced in lysosomes, and keep in mind that plant cells don't have lysosomes. So they hydrolyze macromolecules, and there is one important process happens in the lysosomes, which is autophagy. Autophagy, it's like you have a hoover or a vacuum cleaner in the cell that cleans or remove all the wastes and recycle it. So autophagy, auto from self. So the cell is going to break down something not coming from outside, not a bacteria, not food molecule, no. It will break down its own organic material that is not needed anymore and recycle it. So it allows the cell to renew itself. This is a process that happens in the lysosome by enzymes produced in the lysosome. Peroxisome is a specialized lysosome that only in animal cell as well. It's similar to lysosome. It's membrane bound as other organelles. And its main function is to catalyze reactions that produce H2O2. Peroxisomes are important. Again, they help in detoxifying the cell by removing hydrogen peroxide, which is a toxic substance that can cause toxification in the cell. So peroxisomes break down H2O2, hydrogen peroxide, into water. That's all what you have to know. So it's a specialized lysosome, but it doesn't produce enzymes to break down macromolecules. No, it produces enzymes that break down H2O2, which is a toxic substance. Speaking about vacuoles, and some students might be confused, what's the difference between a vesicle and a vacuole? A vesicle, mainly, it is used to transport and to take material from one part to another, whereas vacuole, it is used for storage. Okay? Large vesicles that stem from ER and Golgi, but mainly they are used for storage. Selective in transport, it means those vacuoles, they have different functions. Their function depends on where they are located. For example, plant cells have large central vacuole. This is where the water and other inorganic ions are stored. We have contractile vacuoles as well. Those are present in bacteria, in unicellular eukaryotic, because bacteria don't have vacuoles. Unicellular eukaryotic cells, they are needed to allow water to move in and out. We have food vacuoles as well. They are formed by phagocytosis. So vacuoles, their main function is to store material in all plant, in all cell types. So let's continue moving on into the last two organelles in this PowerPoint, which are the mitochondria and chloroplast. Now, the first thing we need to start with is endosymbiotic theory. Endosymbiotic theory, it's an explanation for the origin of mitochondria and chloroplast. Mitochondria and chloroplast was or has given an interest for biologists long time ago, because when they looked at mitochondria and chloroplast, they found out that they have double membranes. They have their own ribosomes. They have their own DNA. But this DNA is not needed, because actually the cell doesn't use this DNA in order to do gene expression. It uses the DNA from the nucleus. So they ask a question: why mitochondria and chloroplast have their own DNA? Why they have double membrane? Why they have ribosomes? Why they have proteins? Why they can replicate by themselves? So after analyzing those two organelles, they came up with a theory. It means a scientific explanation. What was the scientific explanation behind having all of those? The scientific explanation was endosymbiosis. What is endosymbiosis? Endosymbiosis is when a cell or a part become embedded in another part, and both of them, they get benefit. So according to them, the endosymbiotic theory states that an early eukaryotic cell engulfed a prokaryotic cell. So an early eukaryotic cell, which is the cell that exists now, but before, it engulfed, like phagocytosis, it took in a prokaryotic cell, and this prokaryotic cell was the mitochondria and the chloroplast. So in other words, the mitochondria and the chloroplasts were prokaryotic cells that were living alone, having their own DNA, having their own ribosome, having double membrane. Then they were incorporated into a eukaryotic cell, and they became since then a part of that cell. But still they have their own DNA, but the cell doesn't need that because the cell already has its own DNA in the nucleus. So they were prokaryotic cell and became an endosymbiotic. It means living in another cell. Now, if you want to state any theory, you need to show some pieces of evidence. How did the scientists prove or tried to prove their theory? By speaking about those 4 pieces of evidence. The first one, that mitochondria and chloroplasts, they have double membrane, and prokaryotic cells have double membrane. They have membrane and they have cell wall, so they have double membrane. They have their own ribosomes, and they have circular DNA, and they are capable of functioning on their own. So the mitochondria, since they have their own DNA and ribosome, they can function by their own, they can produce their own enzymes. Those 4 pieces of evidence, you have to know about those, because those will but will support the endosymbiotic theory. Without those, we cannot say that mitochondria and chloroplasts were alone. But because of those points, they state the endosymbiotic theory. So speaking about mitochondria, it has a double membrane. The outer membrane is a regular membrane, but in the inner membrane, it has folds called cristia. Those folds play an important role in increasing the surface area. And this increase in surface area will allow the function of the mitochondria to be more efficient. Okay, we will come into the cellular respiration later, but as you know, mitochondria, it's the powerhouse of the cell. It's where cellular respiration takes place in order to produce ATP, and keeping in your mind that inside, as we said, there is DNA and there is ribosomes. So a cell that needs a lot of energy, like for example a muscle cell, is expected to have a high number of mitochondria, because more mitochondria, more ATP, more energy. Now when it comes to the chloroplast, we have some similar concepts as for mitochondria, because chloroplast is involved in creating energy for the cell, but this is in plant cell or in autotrophs that cannot eat. So they are going to convert chemical or light energy into chemical energy. Okay, through photosynthesis or chemosynthesis. Speaking about plant cells, the chloroplast, it's the site of photosynthesis where light is captured inside the chloroplast, mainly inside thylakoids. Here there are pigments called chlorophyll. The pigments will absorb the light and convert it into chemical energy. That can be used to make sugar and to make organic food for the plant. So the same thing, chloroplasts, they have their own DNA, and they have their own ribosomes, and they have double membrane. They have DNA, ribosomes, they have their own DNA. And it's the site of Calvin cycle, which is a part of the photosynthesis that we are going to discuss later.